So thanks to Fletcher Halliday for coming. EID spans this wide breadth from like human epidemiology to natural systems, from genetics and microbiomes to epidemiology and population and community ecology. And so what we've been trying to do is highlight speakers that sort of span that breadth of material. And so today we're excited to have Fletcher on to give us some give us a talk about plants and co-infections, which we haven't had either one of those yet in our lineup. So it's a great hole to fill. Fletcher's an assistant professor at Oregon State University. He's in the Department of Botany and Plant Pathology. He went to UNC Chapel Hill for his PhD and then did a couple different postdocs, if I recall. Where were the postdocs, Fletcher? You were in Zurich for a period of time, and then
I was with Jason Rohr at South Florida first. Yeah.
yeah, um, and has done a range of different work on environmental change, biodiversity, and disease relationships. And now I think is doing a lot of work on co-infection. So we're happy to have you here, Fletcher. And with that, I'll kick it over to you. Thanks so much. If people want to like turn off their video cameras, that's fine. If you want to keep it on, that's fine too. But then we'll have a little bit of Q&A, and Will's gonna run the Q&A session at the end and then take you out. Thanks, Fletcher.
Thank you. Yeah, thanks Paul and Will for coordinating this. It's really exciting to be here. I see some familiar faces and a lot of new faces in the audience. I'm really excited to talk to you about some of the research we're doing in my lab. These are pictures, not anymore. These were pictures. Oh my God. These are pictures of one of my favorite plants. This is orchard grass. It's Dactylus glomerata. Here it is growing in coastal Oregon, and over here it's growing in southeastern Switzerland in the mountains. And it's this really remarkable plant that shows huge plasticity and variation in the expression of its physiological traits. And I'm going to talk about that at the end. It's also infected by a really wide variety of pathogens. So this is like a square centimeter of leaf right here. And there are at least three different fungal pathogens infecting this square centimeter of leaf. And so this is a great system to try to understand how parasites and pathogens interact with each other when they co-infect the same host tissue. And that's the topic that I want to talk about today. I want to really briefly introduce my lab. Um, so this is my lab group. Most of the work that was done by Nicole, a postdoc in the group, and Morgan and Quinn, who are undergrads in the group. But none of this research is possible without all the people in the lab, like Aram and Nicola and Bella and Coop and Hannah and Julian. And so I want to acknowledge the contributions that they've all made to this work as well. Research in our lab is pretty broad. We're mostly community ecologists, and we focus on plant hosts mostly and study plant and pathogen diversity and interactions across lots of different scales. And most of the work right now in the lab is really focused on grass hosts, but I haven't actually always worked on plants. In fact, this is the system where I got my start as a ecologist. On the left here, this is Belinus lagoon. And on the right is one of my very favorite parasites. This is a trematode called Euhaplorchus californiensis. And I think it's just the most adorable little parasite. It has these cute little eye spots and a little snout and a frilly little tail. I started working in the system because I was really curious about how parasites interact with each other when they share the same hosts and how host and parasite diversity impact transmission. And this system just has such a rich history when it comes to understanding the factors that influence parasite community assembly. There was this explosion of research in this system of these trematodes that infect this marine snail as their first intermediate host. Um, and the research uncovered competitive hierarchies and interspecific predatory antagonism and resource competition and competition colonization trade-offs. And it was just an amazing introduction for me into the world of plant or of disease ecology. But yet, working in this system, there were some things that I didn't find entirely satisfying. Um, so, one of the problems that I had working in this system was that these snail hosts, they move around all the time, and they're always getting eaten. And the trematodes, they're not just infecting snails, they're also infecting birds, and the birds move around too. And in fact, at Bolinas Lagoon, they're only present for a short period of each year. Infections were really difficult to quantify in the system non-destructively. You can take infected snails and rear them in the lab and get them to shed their cercaria. This is a cercaria. Um, but but these are marine organisms, and having marine organisms in the lab, it has its own set of challenges. And, as a vegetarian, I wasn't entirely thrilled about destructively harvesting thousands of snails and then just sort of depositing their bodies in the trash when I had sort of uncovered what parasites were infecting them. At the same time that I was working in this system, I was also assisting a graduate student in this system. So this is the Point Reyes National Seashore in California, and this is a grassland system. And it didn't take very long for me to discover that this system might be a nice way to overcome some of those limitations. Um, It turns out these plant systems are a wonderful model for understanding disease ecology. We can study plants and plant communities at very large spatial and temporal scales, and there's been a lot of this done throughout the history of ecology as a field. They're incredibly tractable experimentally. We can build whole host communities. We can inoculate them with relative ease. We can place sentinel hosts into the environment and go away. And when we come back, they are exactly where we put them. Our recapture rates are near 100% in these studies when we do mark recapture surveys. And diseases are really readily quantified non-destructively in the system. We can look at a leaf like this little leaf over here, and we know something about who's infecting that leaf and how severe that infection is. And this means that we can go back to the same host again and again and again and repeatedly measure disease. And that's going to be really useful for the kind of results that I'm going to show today. Okay. So off I went to North Carolina to pursue a PhD in disease ecology with Charles Mitchell. And one of the first experiments that I carried out on this topic of co-infection was a detached leaf assay. And I did this with an undergrad named Marcus Lee. And so what Marcus and I did was we took leaves of a grass. This is Avena sativa. These are oats. And we put them on this petri dish here with water agar, and we inoculated them with a drop of water containing spores of a fungal pathogen, Fusarium graminearum. So this little circle here, those are that's a drop of water containing pathogen spores, and you can see this sort of yellowing coming out of this drop of water. That's the fungus growing in the host. That's a lesion that's growing on that leaf. And we tracked the growth of these lesions over time on these detached leaves. And so here I'm showing the days after we inoculated the leaf, and each of these lines is a different lesion. And you can see it's growing at some rate over time. We repeated this experiment. So these are leaves that were previously healthy. We repeated this experiment with leaves that were previously infected with a systemic virus called barley yellow dwarf virus, PAV. And you can see here that this drop of water, there's no lesion growing out of it. These pictures were taken the same number of days after inoculation. And when we look at the sort of rate at which these infections are growing, it's much slower. Okay, and so this tells us that something about this virus is preventing severe infections by this fungal pathogen. Results like this are super exciting for people like me, especially people who are working in plants, because they suggest that there might be certain microbes or organisms that we can inoculate hosts with that might protect them from disease. And so I got really interested in this idea of antagonistic interactions and whether we could unravel them in nature. And there are some huge limitations to an experiment like this, right? These plants, we have an anti-senescing agent in the water agar that the leaves are on. That's so that the leaves think they're still on a plant, but of course that's going to interact with the plant immune system. And so there are lots of factors in here that aren't totally natural. And so to make these comparisons a little more natural, we started developing a model system where we could go out into the field and actually observe within host interactions in real time in the field. And to do that, we started using this host grass. This is tall fescue. It's it's a cool season grass. It's widely naturalized for the production of hay and turf, and it's kind of everywhere in the eastern US. And it's a great model for studying disease and co-infections in particular, because it's commonly infected by a wide variety of pathogens. So this is one of the pathogens that commonly infects it, or parasites, as I also will refer to them as. This is Calototricum cereale. It causes a disease called anthracnose. It's also infected with this one called Rhizoctonia solani. This causes a disease that we call brown patch disease. And then there's another common parasite or pathogen in this system, Puccinia coronata, which causes a disease called crown rust. And so what we did in this system was we started using sentinel plants to track infections on leaves over time. And then we took those temporal data to try and infer something about within host interactions among all of these, well, these three co-infecting parasites. And here's what that data looks like. So I'm showing you the risk of a leaf becoming infected with Puccinia over time from our field collected data on these longitudinally surveyed leaves. So we track every leaf over time. And what you can see here is that as that leaf ages and it's exposed to more parasite propagules in its environment, its risk of ultimately becoming infected increases as well. Okay, so this is for a leaf that hasn't been previously infected by any other parasite. This is what the risk of infection by Puccinia looks like if that leaf has been previously infected by Calototricum cereale. And what you can see here is we get a reduction in risk, just like we did in our detached leaf assay. So something about being previously infected by Calototricum is reducing the risk that this leaf becomes subsequently infected by Puccinia. Okay, we did this for all these possible combinations. We also put hosts out into the field at different times during the growing season. And what we uncovered in this system was that parasites, they're not just competing with one another, but they're also facilitating one another. So this red club here is showing that antagonistic interaction that we saw, but these blue arrows are showing facilitative interactions where the presence of one parasite actually increased the risk of infection or the severity of infection by another parasite. And often, whether we saw facilitation or antagonism was determined by who showed up on a leaf first. So it was impacted by infection sequence. Okay, so this was my first sort of exposure to the sort of reality that despite antagonistic interactions being really well studied and talked about, there are a lot of facilitative interactions that are occurring in nature as well. During my postdoc with Annalisa Leina, I was really lucky to be involved in a collaboration trying to explore some host-mediated mechanisms of these within-host interactions. And in particular, I got to collaborate on this project based on an experiment that Rachel Penchakowski had carried out, where she took this host plant, this is Plantego lanceolata, and she inoculated an individual leaf on a single plant with a powdery mildew fungus. And then she covered that leaf with a mesh bag so that the mildew couldn't spread off of that leaf. It couldn't spread to other leaves on this plant, and it couldn't spread onto any other plants. She compared that to a plant that was mock inoculated and had just a mesh bag placed over its leaf. And then she exposed all of these plants to a variety of different powdery mildew strains. And what we found in this study was that prior infection by one strain almost universally increased susceptibility to other strains. And that this had really broad implications for how strain diversity became spatially and temporally distributed during epidemics. Our best explanation for this is that infection, this initial infection, is modifying the host immune system in some way that's making those hosts more susceptible to subsequent infection by other strains. And Rachel referred to this as once infected, twice infected. So once you get infected, your chance of getting infected again skyrockets. I loved that, which is why I used it for the title of this talk. Okay, so here's two lines of evidence that facilitation is happening quite commonly within these co-infected plant hosts. But I, I'm, I love to work on plants. I think they're an incredible model system, but in my heart, like when I think about who I am, I think of myself as a disease ecologist first, as a disease ecologist who happens to work on plants rather than as a plant disease ecologist. And so when I was working with Jason, I took this little sort of side quest where I started collecting other longitudinal studies of hosts where they surveyed parasite infections in those hosts. And I tried to get as many studies as I could. And I should say, I'm going to present some results from this. They're not published. I think you should interpret them with some degree of skepticism. This is not a true meta-analysis. There is some system here. So Andy Fenton and colleagues wrote a paper describing a methodology for inferring interactions from longitudinal data. I looked at every paper that they cited and every paper that cited that paper. I reached out to all of those authors and asked them, will you share your data with me? And then I asked them, do you know anyone else who would share their data with me? And from this sort of snowballing effort, I ultimately ended up with 13 different data sets from 26 contributors representing over 100 parasite species, 15 host species, and 25,000 observations. All of these studies have two things in common. So first, they're all exploring infection by more than one parasite. And second, they track host individuals over time. And so that allows me to draw some inference about how parasites are interacting within these hosts. And so in each of these studies, for every pair of parasites, I fit the same models that I just showed you for tall fescue. And I use those models to infer the presence of within-host interactions. And then I performed a hierarchical meta-analysis on the hazard ratios that were produced from those models to try and understand if there's a general pattern of within-host interactions. So here I'm showing you the raw data that I put into that hierarchical meta-analysis. And again, I just want to be very specific. The statistical tool I'm using here is a meta-analysis, but I do not have the power to talk about biases in the literature or characteristics of individual studies that you might be able to do in a traditional meta-analysis. So this is not a traditional meta-analysis. Okay, so every point here is a relationship between two parasites from this model that's testing the risk of infection by a later parasite as a function of whether that host was previously infected by the other parasite. The big circles have more data in them, either because there were more hosts in a study or because hosts were surveyed more frequently over the course of their study. And the smaller points are being weighted less because they have less data associated with them. And what you can see just by looking at this is that there's this huge variation in effect sizes here. We have some hugely negative interactions happening. This is antagonism, or I would interpret this as some evidence of competition happening within hosts. And then we also have really, really massive facilitative interactions showing up here. So really positive associations between co-infecting parasites. And when we look at the average across all of these different studies, we see that it's well above zero here. So our on average, we can sort of This analysis at least suggests that these within-host interactions are commonly facilitative, at least across these data sets, across these hosts that I have data for. Okay, so this brings up a question of why we keep finding all of this facilitation in these within-host environments. And a question that's become a really central focus of my lab here at Oregon State is whether facilitative interactions are disproportionately common within hosts, and whether we can unravel the ecological mechanisms that might be driving these within host interactions, that might be driving within host facilitation. So to address this question, one of the first projects that we started out on was a collaborative project between Bella Stiver, who's a PhD student in my lab, and two microbiologists. So Elle Barnes, who studies amphibian microbiomes, and Miriam Ojima, who studies the infant gut microbiome. And we got together and started talking about what ecological mechanisms could be driving within host facilitation. And these discussions ultimately led to the publication of a really short review article that we published in Tree in 2024. And so in this paper, we try to start as simply as possible. And we start by thinking about a fundamental ecological concept, trying to understand where you tend to see facilitation in nature. And what we argue is that facilitation commonly occurs under two conditions. So first, when species modify the environment for one another, they generate new niches that other species can exploit. And second, as environmental conditions become increasingly stressful, this sort of generates more opportunities for these niche modifying taxa to facilitate one another by ameliorating those stressors. And so together, these two processes can generate facilitative interactions and drive community assembly. So here's an example of what I'm describing here in the Sonoran Desert. This is a mesquite tree, and it's attenuating the effects of severe arid conditions in this desert, which facilitates the establishment of these saguaro cacti. We often refer to these mesquite trees as nurse plants. This same process also happens in harsh alpine environments where these cushion-forming nurse plants will attenuate the effects of severe alpine conditions and facilitate alpine species that they'd otherwise compete with. Okay, and some people refer to this relationship as the stress gradient hypothesis. Okay, so based on this idea, we tried to identify specific factors that could be leading us to see so much facilitation within hosts. And what we argue in the paper then is that, well, by necessity, symbionts almost always modify their hosts. In other words, niche modification, it's a requirement for successful infection. And we sort of categorize three broad ways that they do this. They can do this by physically changing their hosts. They can do this by changing the chemical composition of host-associated resources. And they can do this by biologically modifying their hosts, for example, by interacting with host immunity. Moreover, we argue in the paper that hosts are incredibly stressful environments. Every host has some form of barrier resistance. We have skin, plants have a waxy cuticle, parasites and other symbionts, they have to overcome that barrier resistance just to get into a host. Hosts have immune systems that are actively fighting off invaders. This is not just true in animal hosts, it's true in plants as well. And hosts have a limited lifespan. They represent an ephemeral habitat for symbionts. Symbionts have to complete at least one stage of their life cycle and get out of their host before the host tissue dies. Okay, and so these create really important stressors that symbionts, and in our case today, parasites, have to interact with in order to successfully reproduce and have population growth. Okay, so this is, like I said, a really, really short paper, and buried in the supplement of this paper is this huge table that I just want to show you all. And what we've done in this is we found examples of each of these mechanisms that we propose in a huge variety of systems, from mutualists to parasites, from plants to invertebrate hosts to vertebrate hosts. And what I really want to highlight here is that these mechanisms that we're identifying, they're showing up again and again and again. And so there seems to be really strong evidence that these are important processes that play out within hosts in general. Okay, so from these ideas then, I got really excited about the possibility of maybe generating and testing some specific hypotheses. And so one hypothesis that we might formulate is that if niche modification is a key mechanism for facilitation, and modification is required for successful infection, then we might hypothesize that within host facilitation should be more common when parasites share the same ecological niche. And so we turned to our meta-analysis data set, this data set that we have for these 15 different host species. And we looked at whether the similarities, parasites that were more similar to each other, experienced stronger facilitation. And what we find across these three different metrics of niche overlap is that more similar parasites are experiencing stronger facilitation. So parasites that are the same type, that are both macroparasites or both microparasites, experience more facilitation on average. Same when they share the same infection site, if they're both gastrointestinal or both circulatory, and same if they share the same transmission mode, if they're both transmitted through direct contact, for example, or if they're both vectored. Okay, so now we've actually found some evidence, at least from these observational surveys, in support of this hypothesis, which I'm really excited about. We can also generate a hypothesis from this pathway, from this set of mechanisms. If niche modification more commonly leads to facilitation in stressful environments, and hosts are stressful because they're difficult to infect, they're difficult to replicate within, and they're difficult to get out of, then we might hypothesize that facilitation will be stronger in lower quality hosts, in the hosts that are worse at supporting parasites. Okay, before I show you the results of the test of this hypothesis, I should probably clarify what I mean when I say host quality here. And so I'm using this term host quality to describe a property, any property of a host that leads to successful transmission of parasites. We can think of this as the contribution of a host to parasite fitness. This term, host quality, it was really popularized by Felicia Kiesing and Rick Ostfeld, who are using this to describe or explain why some communities, particularly communities of high species richness, often dilute pathogen transmission while others amplify it. It's also drawing really heavily on ideas from Cynthia Downs and Tara Stewart Merrill, who've published really extensively on host competence as a key trait that controls pathogen transmission. And we recently sort of operationalized a definition of host quality in this tree paper that came out just a couple months ago. And so what we argue in terms of host quality is that hosts that are harder for parasites to establish in, that are less exposed to parasites, that reduce the growth of parasites, or that are harder for parasites to get out of, are low quality hosts. They're bad at spreading disease. And on the other side, you have high quality hosts that are really good at spreading disease. And so we might argue then that these low quality hosts, for whatever reason, for whatever mechanism that's playing out here in these boxes, these low quality hosts are more stressful environments for parasites because parasites have a harder time completing their life cycle in those hosts. Okay, so to test this hypothesis with our data, I took a subset of the data. We're just looking at non-human mammals here. And we're doing that because I think it makes a lot more sense to make a comparison across at least some relatively similar hosts than also including the one plant system and the one bird system and the one invertebrate system that we have in this study. And what we see here, oh, right. And so to assess host quality, what I've done is for each host species across all the parasites that infect that host, I've just computed the host incidence rate, the rate at which that host on average, acquires new infections in the studies that were carried out. This is totally mechanism free, right? I'm not saying why, for example, these mouse lemurs are such low quality hosts and these capybaras are such high quality hosts. But what it does is it allows me to sort of categorize hosts based on how frequently they become infected in their relative systems. And what we can see is that on average, these lower quality hosts, they're experiencing much greater levels of facilitation than our high quality hosts. And so that's in support of this hypothesis then. Now, there could be any number of reasons that this is happening. I think we should be really careful in interpreting a figure like this because it is totally correlative. There's one sort of benefit to this analysis, for me at least, which is there was always a possibility that these longitudinal surveys, that the signal that I'm calling facilitation was actually just driven by something else, that a good host at getting infected once was a good host at getting infected twice. And at least this figure seems to indicate that, well, we don't know about the individual level, but at least at the host species level, that doesn't seem to be the case. Instead, the exact opposite is happening. The hosts that are least likely to get infected over here are the ones experiencing the greatest levels of facilitation. Okay, the last thing we can do, so our hypothesis isn't just that low quality hosts experience more facilitation and that facilitation is common when there's high niche overlap, but it's actually that there should be an interaction here, that we should see facilitation only when there's high niche overlap and only in these low quality hosts. And so we can test that with our data as well. And in general, we find pretty strong support for that as well. So here, the blue line is showing hosts or parasites that share the same infection site, and the orange are parasites that have different infection sites. And we can see that that negative relationship only emerges for these parasites that share the same infection site. And we see the same thing for parasites that share the same transmission mode. Now, again, we're limited by the data that we have here and the systems that we have here, and we're comparing across completely different systems. And so there's some potential weirdness happening here, right? We don't have data on different infection sites in these mouse lemurs that are over here or in the capybara that's over here. And so that could be contributing to this result to some extent. Nevertheless, I think these results are really exciting because one, they show really strong evidence that within host interactions are commonly facilitative, or at least parasites are commonly positively associated with one another throughout these longitudinal surveys. And this is, at least to my knowledge, the first sort of comprehensive survey that suggests that facilitation is commonly explained by niche overlap among parasites and these host incidence rates, whatever's driving those host incidence rates. But, there's something not totally satisfying about this. This is not predictive. We don't know why these incidence rates are different across these different species. We don't know if it's because of the system that they're being studied in or some biological property of these different host species, or if it's the different parasite assemblages that infect them. And so the next project I want to share with you is a project that's ongoing in our lab where we're asking whether we can actually predict within host facilitation. And we're doing this using a group of host physiological traits that are linked to life history strategies that we think are also linked to host quality. And this is based on a large body of research dating back at least to the 1980s that suggests that hosts that can be characterized by a slow pace of life tend to be really low quality hosts with respect to parasite transmission compared to hosts with a fast pace of life. And we have all these different traits that we can measure on these plants that characterize their life history strategies and seem to be associated with host quality. Okay, these same life history strategies they often explain how species respond to their abiotic environment. So what I'm showing you here are the results from a survey across a 1,000-meter elevation gradient. Each of these points is a different plant community. And you can see, I'm reporting this physiological trait specific leaf area, which we can sort of interpret as a measurement of host community pace of life. And you can see that as communities become warmer, as you move down the mountain, the community level pace of life increases pretty predictably. And what we've shown in this system is that this same community weighted mean pace of life also predicts disease in this system. And so these traits give us an opportunity to sort of predict host quality, both with respect to how it responds to the abiotic and biotic environment, but also what it's doing to disease risk at whole community levels as well as individual levels, which I'm not showing here. Okay, and so what we're excited to do then is to take this idea of host pace of life and sort of extrapolate it to think about these host associated stressors for parasites. So if pace of life represents variation in host quality, does that mean that these same life history strategies and host quality might predict how stressful an environment is for parasites? And if so, does that mean that it can also predict the strength of within host facilitation? To test that, we've begun using orchard grass as this model system in our lab. Orchard grass is great. It's globally distributed. It has huge amounts of variation in the expression of its physiology and physiological traits. Here it is in the Alps in Switzerland. Here it is in a damp coastal grassland in Oregon, and here it is in this arid environment in New Zealand. It is commonly infected by a huge variety of parasites, and by working on this single host species with tremendous variation in its physiology, we can get rid of a lot of the problems that are associated with comparing different assemblages of hosts and assemblages of parasites that we had in our meta-analysis. We also have found parasites in the system that can facilitate one another. So this is a plant that Morgan and Quinn inoculated with a fungal pathogen called Rhencobrunera orthospora. And just like we did in those detached leaf assays, we can track we can track the severity of infection over time on this plant. And we can do the same thing for a plant that's been previously infected by a different pathogen. This is another rust pathogen, Puccinia striiformis. These two plants are genetically identical to one another. The only difference is that these plants down here were inoculated with this rust 3 days before we inoculated them with the scald pathogen with Rhencobrunera. And you can see that the severity of scald infection is much higher for these rust inoculated plants than these scald only plants. And so that's evidence of facilitation, at least in this system. Okay, so we carried out a small experiment in our controlled environment chamber at Oregon State last spring to try and test this idea about host-associated stressors. We took populations of orchard grass from all across the world. We grew them in this very common environment. Half of them were inoculated with rust, and half of them were mock inoculated with rust. We waited 3 days, and then everyone was inoculated with our scald pathogen, with this Rhencobrunera pathogen. And the first thing we see when we look at our data is that just as we saw looking at whole communities in Switzerland, we see that our key functional trait that underlies host pace of life responds quite strongly to abiotic conditions. Here, all these plants are growing in a common environment. This is actually showing that plants that are adapted to wetter environments have a slower pace of life than plants that are adapted to drier environments. So this in itself is pretty cool. When we look at disease in these plants, we see that our trait is a good predictor of host quality. So here I'm showing the rust-free plants. We're looking at disease burden on these plants in our experiment, and we can see that it increases as a function of host-specific leaf area. And here's what we see when we've previously inoculated these plants with the rust pathogen. The effect is completely different. There's no longer any relationship between specific leaf area and scald disease burden. So Well, and this facilitative effect seems to be happening much stronger in these slow-paced hosts, which are being brought up to match the disease in the faster-paced hosts. We might describe this as within host facilitation, effectively equalizing host quality across these different populations of hosts. Here's another way to look at that result. So here I'm showing you the effect of that rusk inoculation on the severity of our scald pathogen. And you can see that it's much greater than zero. It's facilitative in these slow-paced hosts with low specific leaf area. And that relationship weakens and ultimately disappears as host-specific leaf area increases. Okay, and so again, now we have some experimental evidence in support of this hypothesis as well. So we have experimental evidence now suggesting that as hosts become more stressful, we see stronger, higher levels of facilitation in those hosts. Okay, what about this one? Well, this part's really interesting too. And while we don't have any direct measurements of niche modification yet, we do have some cool indirect evidence that this is happening that I wanted to share. So these results come from a big project that was led by Nicole, where she and Hannah and Coop and Julian and Morgan painstakingly expanded our pilot experiment to include more than 800 host plants and some additional experimental treatments. Only there was one small difference in this experiment. In this experiment, all the rust spores were inactivated. They were all dead. And so what we can test with this experiment is whether the results, these facilitative interactions, are due to just a host response to having a pathogen on it, or whether they're actually due to some change in the host or some change that's occurring as a function of this active live pathogen. And what we see when we sort of prime the host immune system with these inactive rust spores is the opposite response that we saw when we inoculated hosts with a live rust. So what we see here is that we actually get a reduction in disease caused by our scald pathogen on hosts that were previously inoculated with dead rust spores. Okay, and this indicates then that something about having a rust spore on you actually activates your immune system. And that means that that facilitative interaction that we're observing has to be so great that it's actually overcoming that priming effect that's happening. So it really does give us some insight that niche modification must be happening for that facilitative interaction to occur. The last really cool thing that we see here is that this priming effect, it's doing the opposite of what the facilitative interaction is happening. In effect, we're seeing a reduction in quality for these highest quality hosts, and that's equalizing quality in a completely different way when we prime the host immune system with these inactive rust spores. Okay, so to summarize all this, in our orchard grass system, we've observed strong facilitative interactions between co-infecting pathogens. And we've seen this also in tall fescue in North Carolina. We've seen it in Plantago lanceolata in Finland. We've seen it across this huge variety of host species through these observational studies. And we're proposing that this facilitation is predictable and that it results from a combination of host-associated stressors and niche-modifying behaviors of symbionts. In the field, we've shown that biotic and abiotic context can be connected to disease via life history strategies that underlie host quality. And now we're trying to integrate all of these ideas together and make predictions about within host facilitation. And in particular, what I'm most excited about is this idea that host functional traits might provide a nexus for predicting these within host interactions. Okay, we're really just at the start here. We have so many more questions about within host interactions moving forward. In this paper, we identified these four different key components of host quality. And I'm really interested in understanding what role each of these components is playing mechanistically in these within host interactions. I'm really curious about the consequences of these interactions for host fitness, Often when you have synergistic interactions happening within a host, it causes a decline in host fitness that can be greater than the decline in fitness for any single infection. And that leads to a lot of questions about the consequences of these facilitative interactions within hosts on pathogen population dynamics. For example, in African buffalo, we know that within host facilitation can lead to population level interference competition as you get higher levels of host mortality and under co-infection. Okay, so with that, I want to acknowledge all the people and funding sources that have made all of this research possible. I want to thank all of you, and I'm happy to take questions.
Great. Thank you so much, Fletcher. If you have a question, please go ahead and turn on your camera and unmute yourself, and we can just go one by one. Also, feel free to type it in the chat if your audio is not working or if you're just shy. Maybe Andrew, do you want to go first?
Hi. Yeah. Thank you. That was super cool. That was super interesting. Thank you. The thing that struck me, you're thinking about this previous inoculation with the rust, right? And you're saying that is evidence for facilitation. How do you disentangle? Just that it's stressful having an infection. So could it just be that actually it's not to do with the fact that it's the rust per se, it's the fact that it's lowering the quality of that plant. And so, for example, if the plant went through a dry spell beforehand, you would get the same result. Does that make sense?
Yeah, it's a great question. So you might argue that a host getting stressed out makes it sicker. And if a previous pathogen stresses out that host, then that makes it likely to get infected by something else, just like any other stress would. I would argue that that's still facilitation, right? Because it's being caused by that early infection. And one thing that I didn't have time to show you today was we paired, so that sort of that priming treatment that we did, we paired that with a heat wave experiment.
Okay.
And in our heat wave experiment, we exposed these plants to a 2-week heat wave that was a little more extreme than a really extreme Pacific Northwest spring heat wave. These pathogens and these plants really like about 16 degrees Celsius. And we grew these plants for 2 weeks at 30 degrees Celsius, and then we inoculated them at 16 degrees with our scald, and we saw a huge reduction in severity. So this heat wave, at least for these plants, is making them much more resistant to infection, not more susceptible. So in this case, I really do feel pretty confident that the rust is doing something specific to this host that's making it more susceptible to infection by the scald. Yeah.
Cool. Thank you. That's great.
Yeah, thanks, Andrew.
Erin, I think you're next.
Okay, thanks, Fletcher. That was an amazing talk. I really loved it. What a amazing, seamless story that you've laid out, starting from your undergrad work all the way through your lab. So I thought that was super interesting and a really cool framework for predicting, under which conditions you'll see facilitative interactions between parasites. And I'm wondering if you could layer on an additional level once you understand this level even more, which is that the environment affects both the host and the parasite. So, you might have environmental effects that affect host quality, but also environmental effects that affect, how much parasite inoculum there is or how successful those parasites are at infecting hosts. And that maybe sometimes those facilitative interactions might change based on how differentially beneficial the environment is for the host versus the pathogen. And just, I guess, a kind of a general question, wondering if you've been thinking about those types of interactions as well.
Yeah, I think we've been thinking a lot about sort of how we can do this experimentally, and these experiments blow up really, really quickly. This is a pretty active research question for Bella, who's a PhD student in the lab. So she took on these surveys of 2 elevational gradients in the hosts in Dactylus' native range and in its introduced range, and isolated a bunch of fungal pathogens from across these gradients and plans on doing inoculation experiments across temperature with these symbionts that are also found across temperature. And it's really hard because the climate is filtering which hosts and parasites are present and which traits are present. And then it's also likely to be changing the actual interactions as they play out. And so I think we can decouple these things experimentally, but it's going to certainly take a long time. Yeah.
It's a great model system for those kinds of questions, though.
Yeah, I'm really excited about it.
Great work. Thank you.
Thanks.
Maggie, do you want to go next?
Yeah, hi, thank you. Oh, I might have just frozen. This was an awesome talk. I really enjoyed it. I work on co-infection in African buffalo, actually, so I think about some of these questions a lot. And I'm really curious, thinking about the different ways that parasites might be affected by these within-host interactions. So I know in some cases you were looking at like infection rates, in some cases it was the spread of lesions, things like that. And I'm curious if you have any intuition about when these different aspects of niche overlap and potentially of host quality might tend to affect things like the infection rate of pathogens versus maybe the replication rates or pathology or other things within hosts. And like, it seems super interesting maybe if these aspects of host quality could be connected to those different parasite traits in different ways, but I don't know if that's something that you've already been exploring.
Yeah, I think scaling this is really the big question for us. And I think this has to start with some degree of modeling before we can really start scaling it, maybe experimentally. But you can imagine in our orchard grass system, if more severe infections reduce host fitness, or there's a virulence transmission trade-off of any kind, then the population level outcome for these pathogens is not going to match this within host facilitation. And this, I mean, this becomes this huge semantic problem too, right? Because I'm calling it facilitation because on an individual leaf, being infected by one thing makes you get more infected by the other thing. But in terms of what's happening at the population or with respect to coexistence of different pathogens, I think there's still a lot of questions about how that actually manifests in nature. Because yeah, if you have synergy within a host, that doesn't necessarily mean that you have synergy across a whole population. And maybe how that plays out to some extent will depend on the natural history of the actual system that you're working in. And so maybe this isn't that well suited for the big meta-analysis study that we have because the natural history of every system is so different from every other system. I don't know if that answers your question. I think you're really well suited to address this in the African buffalo system.
Yeah, no, that does answer my question. Thank you. I think this is so cool, and thinking about how it might play out differently across scales, I would say is definitely a feature, not a bug. It's really cool. So thank you.
Thanks.
Do we have any other questions? If not, I have hopefully a quick one. The meta-analysis results that you showed were really exciting to me. And you mentioned kind of at the end that relationship between like your metric of basically the infection rate of a host species on average versus the facilitation as being kind of a counterfactual of what you'd expect if it was just purely a host quality argument. And I didn't fully understand that, so if you could talk a little bit more about that, that'd be great. But then also, like when you're thinking about these longitudinal observational studies, how or like what kind of are you doing or thinking about in terms of like kind of getting to like Aaron Mordecai's question earlier about potential changes in host quality over time that are maybe independent of any given parasite but might be seasonal, developmental, whatever, right? How do you kind of begin to separate that question of host quality, or is that something that like future studies need to be designed to actually interrogate? Just in thinking about retrospective kind of like observational data sets like that.
Yeah, so my hot take here, and like this is very personal for me, my opinion, and I don't want to like extend this to everyone. If you really want to understand within host interactions, you have to do it experimentally. And so, in my opinion, this big survey isn't the way to understand that mechanism. It might be a way to shed light on whether that mechanism is actually playing out, but it's comparative and it's correlative, and I think we need to be really careful in how we interpret it. And so thinking about how host quality might be shifting over time within an individual, I think that's best done experimentally and probably on a case-by-case basis. And at least for me personally, like this is why I work in a system that's very experimentally tractable, because I think that for me, that's the way that I can wrap my head around all of these processes. As far as that counterfactual, the way that I'm thinking about it is that if a host species is really good at getting infected by one thing, there's a great chance it's really good at getting infected by a second thing. And that's going to lead to positive associations between parasites on average, right? If you go out and you get one thing and you're just like, if you're just really promiscuous and you're always hanging out with your buddies and you're always getting contacted and you get contacted once, you might get contacted twice. And what we find is that that facilitative interaction, that positive association, it's not coupled in a positive way with the host infection rate at the species level. And so at the species level, this lets me rule out the possibility that these more high quality species are just getting exposed to more parasites. And so we're seeing sort of positive associations among parasites by chance. And so that's why, so the reason that I've been sitting on this data set for so long is that I've been very anxious about whether this positive association is just being driven by that pattern. And I think to me, and I would love somebody to tell me if I'm wrong before I submit this for publication, but to me, it seems like that kind of suggests that that's not the process that's playing out here. Maybe I am actually capturing within host interactions and not these sort of spurious positive associations among parasites at the species level.
Yeah, no, that makes a lot more sense now, how you're interpreting that line. I guess for some of those studies that you're looking at, there are kind of indices of host quality. They're not like leaf area necessarily, but you might be able to get like white blood cell count at a minimum for some of these types of things. Have you tried incorporating some of that kind of physiological data on the host perspective in terms of parameterizing some of this?
Yeah, I went through a process of categorizing life history strategies for the mammals
Gotcha.
exclusively, using like the Panthera and then the age, I can't remember what it's called, database. And I pulled out a bunch of life history characteristics and I built a composite score, which I've done for plants as well. And it wasn't very predictive. And I could predict the incidence rates with it, but it had no predictive power for these within host interactions. And my guess is, for that system, we've pared it down to 11 host species, and we're really limited in which host species those are and how they sort of vary across this spectrum of life history. And I'm just not entirely convinced that it's capturing enough variation in the right way to be reflective of what's going on, but it doesn't show support that at least the way that I'm categorizing life history now for these mammals is predictive of within host interactions.
Gotcha. Thanks so much.
Yeah, thanks.
I had one last question. Sorry, Will. If you don't mind, also about the meta-analysis results, which I thought were really interesting. You mentioned that Fenton paper about detecting like interspecific interactions from longitudinal data. And I'm curious, are those effect sizes you're presenting? Like, I think I'm thinking of this one thing from the paper where it's like you're looking at infection status with one parasite at a prior capture, predicting the other parasite at the current capture, or are they just like contemporaneous associations between parasites, just like trying to think about what each of those points means on those plots?
Yeah, so the Fenton paper, and again, like I hope I remember it. They modeled, they were actually modeling severity of infection within a host and how the severity of infection increases over time within a host, and whether that severity of infection by one parasite is impacted by the severity in the previous time point by the other parasite. What we did for this analysis, and the analysis, we have both of these analyses in the 2017 Ecology Letters paper that I wrote during my PhD, was we sort of modified so Sandra Telfer had this great study where she was studying interactions within hosts as a presence using presence absence. And what we did was we sort of took her presence absence approach and integrated that with this within host growth approach using survival analysis as a way to do that, so that we can track hosts until they become infected and ask whether the risk of infection is impacted by whether or not they were previously infected by another parasite. So it's kind of taking this approach, this longitudinal approach from the Fenton paper that was looking at severity and applying that to a presence absence data set using a survival analysis. And so what we're asking is the risk. So it's a time series data. It's like there's a time. Yeah, it's a kind of a complicated model, I guess, but it's a time varying survival analysis. So it's asking whether the risk of infection is impacted by whether or not the previous observation of that host had an infection by the other parasite. And we're just doing this pairwise. We're not doing it like with higher order interactions or anything like that.
Gotcha. Yeah, that makes a lot of sense. Thank you.
Thanks.
So we're just past the hour, so please feel free to log off if you have other things to get to. But also just wanted to thank Fletcher again for such a great talk and for the questions that came in after.